Hi, hello, namaste, good evening, a warm welcome to Unacademy. In today's YouTube class, it's related with our strategies for enhancement in food production. That is the chapter. So the name of the chapter, it is strategies for enhancement in food production. So warm welcome to all. Let us have the discussion on this chapter. So here it is strategies for enhancement in food production. Okay. Regarding with our Karnataka CET 2021, we introduced some of the courses. They are as follows. One is a plus subscription, where you will going to get the best plan for our Karnataka CET 2021 aspirants with the following feature, or having 12-month plan, 6-month plan and 1-month plan will be there, and it include following important features. That is a continuous entire theory, MCQ, doubt clearance session, mega quiz for individual chapter, and unlimited access will be there for you with respect to this plus subscription. Use my code KCET Murali and get the 10% off. 10% off with respect to the price value. And second one, it is a comprehensive batch. What is the speciality? It is also coming under plus course, where complete syllabus is covered, tips and tricks by top educators, topic wise practice questions, full length mock test will be there. And here myself, I am taking this batch, and along with me the 3 other respected educators will going to take their topics. And use code KCET Murali and get 10% off regarding this class. We introduced another batch, that is Mestos batch for KCET 2021. So what you have to do is, you have to select Unacademy Learners app from Play Store and install it, and under that search for goal Karnataka CET KCET and search my name Murali PG and use my code KCET Murali to join to the plus course by doing particular payment. Okay, that is one type of course. Another one, along with this, I am taking special classes also. To enter into special class, you have to use my code KCET Murali. Okay. So once again, procedure is same. We have to take the our Unacademy Learners app from the Play Store, then install it, then go search for goal to Karnataka CET. Go to Karnataka CET, let us crack Karnataka CET, go to Karnataka CET, and under that search my name Murali PG, use my code KCET Murali. So if once you strike on that one, you are able to enter into my special class. Special class, it is for free, YouTube class it is for free, and this plus course under comprehensive batch or Mestos batch or plus course, whatever it will be, it is by or through payment. Okay. So now this is a comprehensive batch and best plan for KCET 2021 aspirants. So here we are with the multiple batches launching every month, and 6-month course, dedicated doubt clearance session, and course completion in Kannada language also, because it is Karnataka CET. Okay, here and there we are going to use the Kannada if you are not understood the concept. Okay, and best plan for KCET 2021 aspirants. Then daily T20 free practice test is there. So practice test for Karnataka CET, it is for free. You can enroll your name for this one. It started on November 2nd. Okay, and use it properly, so you can achieve something in your oncoming Karnataka CET. And if you want more, please use the plus course or apply for this one by using my referral code or user code, that is KCET Murali, and get 10% off. Anyhow, you have to install the Unacademy Learners app, then search for goal, under that Karnataka CET, KCET, and use my referral code or use code KCET Murali and subscribe for that plus subscription, and you will get more things from our side, from Unacademy related with these classes. Okay, so champion batch is also there. We started it from 15th of October. You can enroll your name for that one also. So today let us have the strategies for enhancement in food production. That is the chapter what we have to discuss. So Devesh, welcome to the class. Good evening. Namaste. So now let us discuss our question one by one. Under strategies for enhancement in food production is the topic. Okay, I added 30 questions for this one. So first question, expand MOIT, MOAT, expand MOIT. So what is this expand MOIT? It is multiple ovulation embryo transfer, multiple ovulation endosperm transfer, multiple ovulation embryo transfer, multi-ovulation embryo transfer, none of this. So right answer for this question, Devesh, got it? It is the multiple ovulation embryo transfer. So as a result of that, we can increase the herd size of cattle in a short period of time. So that is what application part, and it is required to increase the number of cattle in a short period of time. So as a result, we can overcome the scarcity of milk related aspects. Second question, which among the following is marine fish? If you take aquaculture, so in that also we can apply the strategies, different strategies under aquaculture to increase the output or biomass of aquaculture. So second one here, marine fish, which is the marine fish. So musa, katla, sardine, mackerel, it is the katla. Katla is a marine fish. So right answer is C. Okay. Next question number 3. The process by which crop plants are enriched with a certain desirable nutrient is called as what? Enrichment of crop plant with a certain nutrient to get more biomass or the grains. What is that? Process. What is that? Process. It is biofortification. So right answer is B for third one. Then fourth question, tryptophan and lysine are what? What is meant by tryptophan and lysine? Lipid, carbohydrate, proteins. So here we can change this one. None of the above. So what is this tryptophan and lysine? They are the protein particle, means it is made up of amino acid. So amino acids, okay, tryptophan, lysine, okay, coming under protein concept. Next, fifth one, identify the process of plant breeding from the diagram shown below. Identify the process of plant breeding from the diagram shown below. Here, self-pollination, fertilization, artificial cross-pollination, natural cross-pollination. It is called artificial cross-pollination, where emasculation is the process, isn't it? So as a result, we can remove unwanted anthers or stamens to overcome the pollination. So through artificial way, our desired pollen grains can be dusted on the stigma. So it is controlled pollination mechanism to produce hybrid plant. It's a traditional method, okay, artificial cross pollination. So after emasculation, bagging and tagging is done. So here, emasculation commonly done before the opening of flower, refers to in bud condition. Next, Norman Borlaug or Norman E. Borlaug, the father of Green Revolution, has developed a highly recognized and appreciated cultivating variety of what? Wheat, rice, onion, sugarcane. It is a Norman E. Borlaug developed wheat variety, isn't it? Wheat variety, and it will going to increase the yield. So another question can be done. Who is considered as father of Green Revolution? Norman E. Borlaug. Then, which of the following helps in absorption of phosphorus from soil by plants? Which is the thing? Yes, Kishan, welcome to the class. Good evening. Okay. So which one of the following helps in absorption of phosphorus from soil by plant? Enabina, Rhizobium, Glomus, Frankia. So use of this microorganism in the case of enrichment of soil. So for the absorption of phosphorus, so phosphorus item is not going to be absorbed directly from the plant, directly through root in the case of plants. So one of the organism, the fungal species, it is the Glomus. Glomus species will help in the assimilation of nutrient like phosphorus from the soil. So the plant will going to get the phosphorus, and in terms of that, the Glomus species will going to get the shelter and food from plant. So it is a mutual relationship, between the plant and that microorganism or fungal particle, that is a Glomus. Okay. So right answer is D for seventh one. Then eighth one, selection of homozygous plant is through which method, or what is that? Mixed selection, mass selection, pure line selection, none of this. It is pure line selection. Pure line selection, through which we can have the selection of homozygous plant, where the genetic material will become homozygous. It is not mix of these things. Only the required characters or narrowing of character will be there. Only particular character is going to be highlighted there. So option, it is C for 8th one. Yes, Sheshika. Yes, option is C. Yes, you are right. Sheshika, welcome to the class. Good evening. Next question number 9. An organism used as biofertilizer for raising soybean crop. So microorganism usage with respect to our cultivation of crop, which is that one. So here the right answer for the 9th question, it is rhizobium. Rhizobium, it is utilized as biofertilizer and it is one of the nitrogen fixing microorganism. Okay. Next, 10th question. In tissue culture medium, means in the micropropagation, the embryo is formed from pollen grain is due to which process? Embryos are going to be formed from the pollen grain is due to what? So right answer, it is double fertilization, organogenesis, test tube culture, cellular totipotency. It is the cellular totipotency. Cellular totipotency means single cell is responsible or having the ability to give rise to entire organism, entire plant here in the case of plant tissue culture. So that is what cellular totipotency. So in the case of micropropagation, we are selecting one particular cell which is showing totipotence. So as a result of that, that will going to give the actual entire organism under tissue culture. Next, 11th one. Hybrid vigor is mostly due to what reason? What is the thing behind the production of hybrid vigor? Hybrid plant, hybrid production is because of what? Yes, for question number 11, am I right, Devesh? Hybrid vigor, that is homozygosity of pure character, superiority of all the genes, heterozygosity, mixing up of cytoplasm of male and with that of the female. No, it's not mixing, it is C. It is heterozygosity. We are going to give hybrid vigor, mixing of characters. Then, main objective of production or use of herbicide resistant genetically modified crop is to what? What is the reason behind that one? Yes, Saurav Patil, welcome to the class. Good evening. Devesh, yeah, you are right. For the previous question it is C, and for 12th one it is A. Reduce the herbicide accumulation in food articles for health safety. So production and use of herbicide resistant genetically modified crop, GM plant, which are showing resistant power against herbicide. So as a result of that, we can minimize the use of chemicals to kill that unwanted things. So reduction with respect to herbicide accumulation in food article, because once it is added, it will be transported through the food chain. So to overcome that one, we are having or we are applying that particular phenomena where genetically modified crops which are showing resistance power against particular diseases. So thereby we can reduce the herbicide use. Next, 13th one. In which crops is the method of mass selection applied? Mass selection is applied in the case of cross pollination, pollinated crop. Mass selection is applied. So for 13th one, yeah, 12th one, it is A, that is right, Devesh, good work. And for 13th one, 13th question again, for 13th question, the right answer is A, cross pollinated, where mass selection is going to be applied. Then, plants provide protection from fungal disease by producing what? Plant provide protection from fungal disease by producing which chemical? Phytohormones, phytochemicals, phytoalexin, all of this. Right answer is C, phytoalexin. Yes, Saurabha, you are right. For previous question, it is A. 13th question, it is A is the right answer. Good work. Yeah. For 14th one, the right answer is C is the right answer. C is the right answer. Okay. Yeah. For 14th, C is the right answer. Next, I will move on to the next question. That is 15th. Sterilization of tissue culture medium is done by which method? Sterilization of tissue culture medium. It is by autoclaving of medium at 120 degrees Celsius. Okay, it is not like this. 120 degree Celsius. Right answer is C. 120 degree Celsius. So as a result of that, we can kill the microorganisms which are going to have the contamination. So to avoid the contamination, we are doing sterilization of tissue culture, media, tissue culture the utensils using in the tissue culture, and other surface areas. So that is very much essential. Okay. So 15th question, C is the right answer. Yes, for 15th, C. Shreyas. Okay. Good evening. Then the problem of necrosis and gradual senescence while performing tissue culture can be overcome by gradual senescence, aging of that callus or tissues, and necrosis of that tissue can be overcome in the continuous tissue culture performance. It is by which method? It is subculture. So if you are going to keep the culturing media with that explant or that callus continuously in a single way, it will going to have the necrosis and gradual senescence. So senescence will be there, the death of the tissue or the cell will be there, or we can observe the aging phenomena. So to overcome that aging phenomena, the subcultures are going to be done alternately. Then plant tissue culture media can be solid, semi-solid, liquid, all of this. So 17, right answer, it is D is the right answer. D is the right answer for the question number 17. Then 18th question. The main purpose of using UV radiation in plant tissue culture process. What is the use of UV radiation? So mutation, alkylation, autoclaving, disinfection. It is nothing but disinfection. Okay, it is nothing but disinfection. So to overcome the disinfection, to kill the microorganism, to eradicate the contaminating object, we are doing the UV radiation use. So before the tissue culturing practice in laminar airflow chamber. Then match the following and choose the correct combination. Here I given in the first column honey, bee venom, propolis and beeswax. So economic important byproduct, isn't it? And in the second column I given the use of that one. So right option for this 19th question, it is A. Refers to honey, it is artificial sweetener, bee venom, honey bee venom. So in the case of worker honeybee, we can observe the bee venom, isn't it? So it is utilized in the treatment of rheumatoid arthritis, autoimmune disease. Then propolis, it is antiseptic and antibiotic property. And last one, beeswax, used in the cosmetics and creams. So right answer is A for 19th question. Next 20th one. Okay, 20th question. The best way to obtain bacteria and viruses free plant through tissue culture, by using which one? So shoot apex, or meristem culture. Why? Because meristematic tissue, meristematic cells are free from virus or bacterial attack. So for that purpose, this shoot apex with the meristematic or shoot apex or meristematic cells are going to be utilized under meristem culture. So as a result of that, bacteria and virus free plants can be obtained even though the plant is having bacteria or viral attack. So shoot apex can be utilized. Yes, Saurav Patil, you are right. Shreyas, for previous question, 19th question, A is the right answer. Yes. Because here what happens is the cell division rate of bacteria and virus is lesser than that of cell division rate in the meristematic tissues. So virus and bacteria are not getting enough time for their particular cell division. So the rate of cell division is faster in the case of meristematic region cells than the bacteria and virus. So unable to grow, unable to infect that meristematic region by bacteria and virus. And next question, which one of the following products of apiculture is used in cosmetics and polishes? Apiculture means honeybee byproduct used in cosmetics. So which is that thing? So 21st question, it is the beeswax. You can write beeswax, isn't it? Wax refers to beeswax. So it is utilized in polishes and cosmetics purpose. Yes, Saragappa, welcome to the class. It's not honey, okay? It's not A, honey, it is C, wax. Honey is used in case of medicinal purpose, okay, medical purpose, in Ayurvedic medicine. Then 22nd question, more than 70% of livestock population is in which nation or which country? It is Denmark, India, China, India and China. According to the statistical approach, it is D is the right answer, India and China, livestock population. Okay, that is a cattle, etc. Then the agriculture sector in India employs about how much percentage of population? So agriculture sector in the case of India. So 23, question number 23, am I right? Yeah, it is about 62% of population, directly or indirectly. Okay, yes, Saragappa, you are right. For the previous question, D is the right answer. And for 23rd question, it is once again D is the right answer. Okay, so we are with the 30 questions, we will complete. Okay, so time is less, so don't worry, we will extend for about 5 minutes extra. So 33% of India, that is GDP, gross domestic product, GDP, comes from which sector? Industry, export, small scale cottage industries and agriculture. So 24th question, the right answer is B. From agriculture, about 33% of India's GDP, because in the previous question I said about 62% of population is directly or indirectly employed with the agriculture sector. So that's much of percentage will going to provide about GDP to our nation, 33%. Next, 25th question. A collection of all the alleles of all the genes of a crop plant is called what? What is the technical terminology? It is the germplasm collection. All the alleles of all the genes collection, it is called germplasm collection. So right answer is which one? For 25th, it is A, Saurav Patil. Yes, you are right, germplasm collection. Then 26th question. system of rotating crops with legume or grass pasture to improve soil structure and fertility, it is called what? What is the terminology with respect to that things? So system of rotating crops with legume or grass in between. So pastures of legume and grass to improve soil structure, which is that one. So strip farming, shifting agriculture, lay farming, counter farming. This is the lay farming. Answer is C for 26th. So if you have any doubt, you can ask. Okay, which of the following enhances or induces fusion of protoplast under protoplast culture? Which is the chemical used in the fusion of protoplasm? 27th, it is a polyethylene glycol and sodium nitrate is going to be given in the culture media where protoplast is going to be fused. Yes, for 26th, Shreyas, it is C, you are right. Good work. So polyethylene glycol and sodium nitrate is going to be applied during the time of protoplast fusion. So induction of fusion, so influence of the polyethylene glycol and sodium nitrate will be there for the fusion of protoplast under protoplast culture. Next, a technique of micropropagation is somatic hybridization, somatic embryogenesis, protoplast fusion, embryo rescue. So 28, it is the somatic embryogenesis. It is the technique of micropropagation. 28th answer is B is the right answer. Okay. And next question number 29. The process of induced breeding of fish is called as what? Induced breeding, the first once again the administration of hormone. This will be there for the production of large number of offspring. So 29, it is called hypophysis. So there in our herd community, how to increase the cattle, that is a multiple ovulation embryo transfer. There also influence of hormone is there. Super ovulation methodology, by the administration of hormones to produce the multiple ovule. Okay, so that is the moit. Here in the case of aquaculture, in the fish breeding, so induced breeding can be done by the administration of hormone. It is called hypophysis phenomenon. So for that the right answer is A for 29. And last question, it is who is the father of white revolution in India? Not green revolution, white revolution in India. Green revolution, M.S. Swaminathan. Okay. And the answer for this white revolution related with the milk and milk product, it is Verghese Kurian. Okay, Verghese Kurian, not Nargis Kurian. It is a Verghese Kurian. So who started Amul in Gujarat, isn't it? Yes, Saragappa. For the previous question, I think M.S. Swaminathan, no, Saragappa, it is M.S. Swaminathan for green revolution in India. For white revolution, it is the father of white revolution in India. It is Verghese Kurian. Got it? So this is the last question of this class. And if you want more thing, use this opportunity by subscribing for Plus class, where best plan for Karnataka CET. And here what you have to do is select Unacademy Learners app from the Play Store, install it and search for goal. Under that Karnataka CET, use my name Murali PG or use my code KCET Murali during the subscription. And for special class also, you have to use my code for the entry to my special class category. Okay, KCET Murali is the use code or referral code. So for plus subscription, you have to use this one, and as a result you will get 10% off. Special class is free. Okay, so comprehensive batch is there. So here what is the specialities? Complete syllabus, tips and tricks by top educators, practice questions will be there, mock test will be there. So as a result of that, you will going to enhance, or you will going to get more things from our side, from Unacademy, regarding with the Karnataka CET and our future oncoming the different types of competitive exams. So better if you choose this batches. So Mestos batch, newly introduced batch, it is also coming under this plus course. Okay, use my this my referral code KCET Murali while you are subscribing for this class. Then comprehensive batch, myself also taking the comprehensive batch. Best plan for Karnataka CET. Remember, my dear students, for special class also you have to use this code KCET Murali to enter into my class. Okay, and it is free, but for plus course you have to pay. So payment is very less actually when comparing with other courses. So 12-month plan it is 12,000, 6-month plan it is 10,000. Just imagine the or see the difference. Okay, learners app, Unacademy learners app. Under that, go to search goal, give that one, and under that Karnataka CET, KCET, and use my referral code and enter to the classes. So daily T20 free practice test for Karnataka CET also there. Use this one. Okay. So thank you. See you again in the next class. My next YouTube class, it is on 23rd of this month. The chapter is molecular basis of inheritance. Okay, my next YouTube class is related with the molecular basis of inheritance. Okay, my dear students, good night to all. So we will see again in the next class. So I am taking many special classes. You can join to that one also. Every day at 9 p.m. I am taking it. Tomorrow 9 p.m. I am going to take a special class regarding with the the topic, I think human disease and immunity, human immunity and diseases. That is tomorrow's special class topic. Okay, at 9 p.m. So let us see that one on tomorrow. Okay, thank you one and all. Good night to all. I am going to wind up this class now.